Hello everybody, welcome back to our third and final video review for this exam, Exam 1, covering properties of water, functional groups, and macromolecules. I think I told you guys all in class that you will get 2 FRQs from College Board, which I will not be going over, and then one longer review that I wrote. The reason I had to write this one is one, I did do College Board style, but two, there just aren't a lot of College Board questions that only talk about the topic that we've covered, because we're so early in the year that usually long FRQs require long discussions from all of the units from the whole year. So I had to write my own long review this time. That's why I'm giving you a little bit of a heads up by doing a parallel question in this review. So please pay attention, make sure that you understand all parts of this, and if you struggle to understand anything, send me an email or post in the OSS forum, and you can go ahead and let me know if you're confused anywhere. Okay, I wasn't able to finish everything at school today, so now it's being brought to you from my own home. But let's go ahead and just get started on question 1 here. A team of microbiologists discovered that a specific single-stranded ssDNA DNA virus can cause mutations in the FAB-A gene in E. coli. The FAB-A gene is known to encode the FAB-A enzyme that allows bacteria to convert saturated fatty acids to unsaturated fatty acids and vice versa. Let's pause there. So you might have been immediately shocked by this statement, a specific single-stranded DNA. And you might have thought, well, wait a second, there has to be something wrong. DNA is double-stranded. And you're right for the vast majority of biology, that 99.99999% of the time DNA is going to be double-stranded. However, viruses are a little bit special. They're not cells, and so they actually can have single-stranded DNA genomes. They can also have double-stranded RNA genomes. These are both possible for viruses. So if you see that maybe somewhere on the test, either in this sort of FRQ or in a multiple choice, don't be shocked. Don't just immediately eliminate something that's talking about a virus with single-stranded DNA or double-stranded RNA, okay? All right, now let's go ahead and look at question 1 here. Explain the significance of the FAB-A enzyme in maintaining membrane fluidity and rigidness. So if this FAB-A enzyme is very important for converting between saturated and unsaturated fatty acids, I want you to start thinking about, okay, how does a bacteria take advantage of this enzyme or use this enzyme when maybe conditions change? Think about what or which fatty acids are going to be liquid, which fatty acids are going to be solid at room temperature, and then if temperatures get really, really, really cold, how will the bacteria respond? Meaning, will the FAB-A enzyme convert between saturated and unsaturated? In which direction will it prioritize? Or if it gets really, really hot, which one will it prioritize, saturated or unsaturated? So I want you to be thinking about that. A correct answer would discuss how that enzyme is very important in terms of switching between saturated and unsaturated fatty acids as a form of regulating, I guess it's temperature, right? It's not actually regulating its temperature, it's really responding to the temperature, right? Okay, second one here is just do you know it or not? Identify the monomer of a protein encoded by the FAB-A gene. So hopefully you know that an enzyme is a protein. I was a little generous on this one. But if you know the monomer of a protein, this is an easy point. The second one here, or the third one here, is discuss how a mutation in the bacterial DNA caused by the single-stranded DNA virus could alter the function of the FAB-A enzyme. Okay, so this, I want you to start thinking again about that idea of structure dictates function. Structure dictates function, right? So if we have mutated the DNA codes for RNA, codes for protein. Well, if we've altered that DNA, we could potentially alter that protein. If we have an altered protein, an altered structure, an altered shape, then we are going to have an altered function. So I want you to start putting those pieces together. All right, that's how I would approach question 1. Now, I'm not going to tell you exactly how to answer it, but I do want to get your mind spinning. Okay, so the next couple of questions are all going to be related to the same scenario, so keep that in mind as we move on to the next questions. This question says, researchers are interested in isolating a specific gene from a new plant species and amplifying it using PCR for further analysis. When we say amplifying using PCR, we are just saying that we are making lots and lots and lots of copies of that gene. So we are going to be copying that gene over and over and over and over again, upwards of 1 million times. So that's what we mean by amplify. During PCR, the DNA strands must be separated. We call this the denaturation step. The researchers have a PCR protocol optimized for DNA that contains 30% adenine, 30% thymine, 20% guanine, and 20% cytosine. Upon sequencing the DNA of their gene of interest, they discovered that it contains 40% AT content. Calculate the percentage of guanine in the DNA of interest. Well, if the DNA of interest is 40% AT, that means that it has to be 60% GC. And half of that 60%, because DNA is double-stranded, is going to be guanine, and the other half would be cytosine. So half of 60% is 30% guanine. The second part here is a little trickier. So explain how the researchers might need to adjust their PCR protocol to accommodate this new AT content with reference to the role of hydrogen bonds in DNA stability. So this was when I was talking about, in your notes, I said highlight, 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 star, 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 that there are 2 hydrogen bonds between A's and T's, and there are 3 hydrogen bonds between G's and C's. So if the original protocol, the one that they have optimized, is AT rich, and it is, right? 30% adenine, 30% thymine, that is going to be 60% AT, or just 40% GC. Since it is technically GC poor, that means that we don't need quite as much energy, aka heat, to separate or denature the DNA strands. But because our new gene of interest is 40% AT and 60% GC, well, that means it is GC rich, which means I need to put more energy in order to separate the strands. So during the denaturation step, I'm going to have to raise the temperature. I'm going to have to raise the temperature. And that is a really key idea here. And I really want to make sure that you understand the 2 pieces here. You need to tell me exactly what the scientists should do. Do they raise the temperature? Do they lower it? You need to explain why. And you need to explain the role of hydrogen bonds here. So 3 parts here. Okay. The next question, again, relates to that idea, right? So after PCR, the scientists decide to use their amplified DNA to express the protein of interest to verify that it carries out the function they believe it does. To do this, the scientists apply the necessary transcription factors to induce transcription of the gene. They then allow the cell to translate the transcription product into their protein. So, this might seem like it's beyond the scope of this unit, but it's really not, because I know that most of you guys still remember transcription and translation. And remember, we talked about what is the role of DNA. What does it code for? That is going to be transcription. So DNA to RNA is transcription. RNA to protein is translation. So make sure you know that. The scientists decide to isolate the protein downstream experiments. They find that their standard isolation protocol is only yielding small amounts of protein. The protocol calls for a dissolving step in water, and the scientists predict that this is the step that is causing the low yields. So, The question asked, predict what may be different about this protein if the scientist's prediction is correct. So remember, the scientist's prediction is that the dissolving step is the step that is causing low yields. And identify what feature of the protein monomer that causes that difference. Okay, so what might be different if this protein is not dissolving in water? What might be different about that protein? Well, most proteins are polar, and they are going to be, remember, like dissolves like. So they will dissolve easily in water. So maybe you can make a prediction about what is different about this protein if it can't dissolve in water. The other part of this is, what feature of the protein monomer causes that difference? Well, remember that some parts of the protein monomer stay the same, right? NCC, NCC, that always stays the same. But think about what is different, and if that difference could potentially play a role in whether or not it'll dissolve. So think about those things, maybe pull back that chart with all the different amino acids in it and see what the groups are so that you can adequately answer this question. Okay, so there is one last question on your FRQ that is relating to that scenario, but I'm going to leave that up to you as you are preparing. Maybe try to anticipate what else I could ask dealing with that scenario, but I'm going to leave that up to you to prepare for. I will be very frank that those couple of questions are very similar to what you will see on Monday or Tuesday. So you will be very, very prepared for that FRQ. You will have 2 others from College Board, which I cannot go over because I cannot share College Board FRQs publicly, but I do want you to feel okay about them. They are not too difficult, I promise. Now, as you prepare for the exam, your best tool, again, is going to be that study guide. Don't waste that practice. Really, really try and make sure that you know that. Don't just try to get answers in to get a grade. Yeah, I probably won't notice the difference, but you're wasting your opportunity to get a lot of meaningful practice there. So that's your best tool. These videos are going to be your second best tool, and your third best tool is going to be going through the slides and making sure that you know every single thing on the slides. I've posted them to OSS Connect for you so you can look at them yourself. My recommendation is to pretend to teach it to an audience. I think I talked about this in class a little bit today. So pretend to teach it to a classroom. When you get stuck, that is somewhere where you probably need a little bit more work. You can write it down, go back, and study it. Quizlet is also great. There are definitely slide decks out there for Unit 1. Is it going to be exactly what I covered? Probably not, but there's a lot of overlap. And of course, if you make your own Quizlet and study that, that's excellent as well. With that said, I wish you the very best studying this weekend. Maybe make a day of it and do a watch partying with all these videos. And I hope that you do it super, super well. I'm rooting for all of you, I pinky promise. And I am looking forward to seeing these scores when I go over it on Wednesday. So best of luck, get some rest, and have a great weekend.